For most of Earth's history, the deep ocean was a void, until life began to generate its own illumination. While an incandescent bulb wastes the vast majority of its energy as heat, biological systems channel a high proportion of that energy directly into photons. The result is a process of exceptional energy efficiency. The primary substrate, luciferin, is activated by the catalyst luciferase. When oxygen and ATP enter the system, they react to form a highly volatile, excited state intermediate. Returning to a stable, lower energy level, it releases that energy as visible light. Across nature, organisms use structurally distinct luciferins to achieve this. It is a clear case of convergent evolution, where the ability to glow emerged multiple times, completely independently. Life engineered a specialized chemical technology to navigate and conquer the lightless void. Generating endogenous light is metabolically expensive. For this trait to persist across so many species, it must provide a selective advantage that outweighs the high calorie cost. Predation is a primary driver. The anglerfish, for example, hosts symbiotic bacteria in a specialized lure, using a faint glow to exploit the natural attraction of prey in a lightless environment. Defensive strategies in the dark are equally sophisticated. When disturbed, dinoflagellates emit sudden flashes of light that function like a burglar alarm, exposing their immediate predators to even larger hunters nearby. This geometric model illustrates counter-illumination. By matching the intensity of downwelling light from the surface with specialized organs on their bellies, squid and fish erase their own shadows to hide from predators looking up from below. If spotted, some shrimp use a decoy release, ejecting a cloud of luminous material that provides a false target while the shrimp escapes into the darkness. This light also serves as a signaling protocol. Species like fireflies and marine ostracods use specific intervals and patterns to encode their identity and mating readiness. These flashes are not random. Each pulse is an optically tuned strategy for survival. The color an organism emits is dictated by the specific optical properties of its environment. In the ocean, blue-green light between 470 and 490 nanometers travels the furthest with minimal absorption. Most marine species have evolved to both emit and detect this specific range. On land, the physics shift. Evolution favors yellow-green emissions, roughly 550 to 570 nanometers, to align with the visual sensitivity of terrestrial insects. However, the deep-sea dragonfish circumvents these standard rules to gain a predatory edge. It produces red bioluminescence, a wavelength that is almost entirely invisible to other species in the deep. This creates a private wavelength channel. The dragonfish can illuminate and scan its surroundings while remaining effectively invisible to its prey. Mastering the physics of color allows certain predators to navigate the visual landscape on a channel their prey cannot detect. The process of creating light chemically is completely distinct from how organisms simply transform it optically. On the left side of this graphic is bioluminescence. It requires an external UV or blue light source to function, absorbing those incoming photons and re-emitting them at longer wavelengths. This is a dependent process. If the external light source is removed, the fluorescence ceases instantly. Bioluminescence, however, is endogenous. It is a chemical emission that functions in complete, absolute darkness. When the water violently churns, millions of microscopic organisms instantly react, filling the absolute darkness with brilliant chemical light. Human technology leverages these biological mechanisms. Luciferase-tagged cells visualize tumor growth, and engineered microbes detect environmental toxins. In a cold, lightless world without a sun, life does not wait for the dawn. It generates the light itself.